Glycogenesis. Glycogenesis is the process of glycogen synthesis, in which glucose molecules are added to chains of glycogen for storage. This process is activated during rest periods and also activated by insulin in response to high glucose levels. The key points of glycogenesis: the synthesis of glycogen from glucose is called glycogenesis. It occurs in cytosol. Besides glucose, it refers ATP and UTP. Now let's look at the reaction of the glycogenesis. There are 4 steps of reactions. First is the synthesis of UDP glucose. Second is the requirement of the premier to initiate. Third is the glycogen synthesis by glycogen synthase. And the last one is the formation of branches in the glycogen.
First step of glycogenesis involves the conversion of glucose to glucose 6-phosphate, and the second step is from glucose 6-phosphate to glucose 1-phosphate. Now let's take a closer look of this part. Glucose is absorbed from the intestine that causes our blood sugar to go out. The pancreas responds to high blood sugar by secreting an insulin. Now, the insulin acts as the glucose transporters. This one. And it passes through the glutes. So, it makes them bring more glucose. Yan. More glucose inside the cell in our body. Inside the cell, an enzyme called hexokinase add phosphate group to its 6th carbon, creating the G6P, or the so-called glucose 6-phosphate. So this is how the glucose converted into glucose 6-phosphate. Yes, and here in this step, if the glucose is present in the muscle, the process is catalyzed by the use of the enzyme hexokinase. And if the glucose is present in the liver, the process is catalyzed by the use of this enzyme called glucokinase. And there is also one molecule of ATP is converted into 1 molecule of ADP, or the so-called adenosine. From adenosine triphosphate is converted to adenosine diphosphate. So now let's move on to the second step, which is the conversion of glucose 6-phosphate to glucose 1-phosphate with the use of this enzyme called phosphoglucomutase. So in this step, the glucose 6-phosphate is then rearranged so that the group of phosphate moves from the 6th position down to the 1st position. Of course, with the use, or this reaction can't be done without this enzyme called the phosphoglucomutase. So as what I've said a while ago, the phosphate group will be moved from 6th number 6 position of the carbon down to the number 1 position. So this will be the yan. And here will be OH. Yan. It will be balanced out, creating the glucose 1-phosphate. So that is how the glucose 6-phosphate converted into glucose 6-1-phosphate with the use of the enzyme phosphoglucomutase.
From glucose 1-phosphate and UTP, the present of enzyme is UDP glucose pyrophosphorylase.
To initiate the process of glycolysis, we require R primers, generally the pre-existing small fragments of glycogen, acts as a primer, but in the absence of that, small fragments of glycogen of protein named glycogenin excerpts glucose molecule from UDP and forms a primer. These glycogen initiators in this transfers the first molecule of glucose to glycogenin. This is where glycogen primer is found.
Now, the transfer of glucose molecules from UDPG to the glycogen primer is done by the enzyme glycogen synthase. Thus, this enzyme is responsible for the one-fold of glycosidic bond. Now, the branch formation takes place, which is catalyzed by the enzyme glycosyl alpha 46, which transfer 5 to 8 molecules of glucose from a non-reducing end of glycogen and attaches to another glucose molecules. Thus, the alpha 1 to 6 bond is formed. Now, a simultaneous action of glycogen synthase and glycogen alpha 4 to 6 transferase is observed, and finally, at last, glycogen is synthesized. In conclusion, the inhibition and significance, the glucagon or in the liver and epinephrine, the liver and the muscle, inhibit glycogenesis via the CAMP or the cyclic adenosine monophosphate protein kinase A phosphorylation cascade, which results in phosphorylation and thus the activation of glycogen synthase. The glucose and its precursors are obtained through food. However, under certain conditions, they may not be a reliable and continuous source of energy. The glycogenesis process is therefore a built-in mechanism of the body which stores the excess carbohydrates we consume in the form of glycogen, which could be broken down to glucose when needed.